Welcome to Neurosalience, the OHBM podcast.
Today my guest is Dr. Stephanie Forkel, a Donders principal investigator and assistant professor at the Radboud University studying the impact of neurovariability on cognition in health and disease. In 2013, she received her PhD in neuroimaging at King's College in London. And in this work, she helped establish an understanding that neurovariability is critical for prediction of recovery after stroke. Over her academic career so far, she has continued to develop this line of work and has trained in many different places, including University of Salzburg, National University of Ireland in Galway, Ludwig-Maximilians-Universität in Munich, Klinikum Großhadern in Germany, University of Greenwich in the UK, UC Davis in the US, and CNRS in France. Brains are both similar and different across individuals and species. Understanding how they are similar and how they differ, and how this all ties in with behavior, is not only central to understanding principles of brain organization and evolution, but deeply important for how effectively these brain imaging techniques can be used clinically. I've noticed also that the field of functional MRI has started to move just away a bit from large group averages towards embracing the challenge of understanding individual differences. Dr. Forkel has been working on this in the context of white matter tract mapping since before 2012. Dr. Forkel has been truly a dynamic trailblazer, a thought leader in both basic and clinical neuroimaging, and she's taken on many roles in the Organization for Human Brain Mapping recently, and so is very busy, but doing great work. So I hope you enjoy this podcast. Okay, welcome, Stephanie Forkel, to the OHBM Neurosalience podcast. And, I just wanted to first just mention that I got to know you through the podcast, actually, through OHBM ComCom. And I was so impressed with how you took charge in sort of taking podcasts and putting it up on YouTube so effectively. And I realized that that's sort of indicative of, in general, how you work. And, recently you've been, taking on leadership at the OHBM. Now you're the program chair and, that which oversees the scientific content for the entire meeting. And so on top of that, now I've had a chance to sort of look into your work, which I just find not only really original and rigorous, but also I think really exciting as far as the possibilities it opens up. So before we get into talking a little bit about that, just wanted to briefly start with your history a little bit, looking at your website, and you've been all over the place in terms of your education. You've it seems all over Europe and elsewhere, and you have such a diverse background, in terms of education and training. So just going back further, so where did you grow up, and how did you first become interested in the brain?
That's going back a long time. So I grew up in Munich in the south of Germany. And from an early age on, I got interested in science because my mom was a single mom at the time. So I always had to spend time with her at work, which was in the hospital. So the hospital environment to me was very natural. And then I ventured out to study psychology in Austria. And realized that I needed a bit more brain in my studies. So I did an internship in a neurological clinic back in Munich and worked with patients with neurodegenerative disorders and stroke. And I really, really enjoyed studying the brain in that context. So I asked the medics if I could shadow and observe their work a bit more. And the only medics who said, of course, if you want to join us, you're more than welcome, were the pathologists. Because they're usually not used to people voluntarily coming to join them. And that's how I got into brain anatomy and stuck with it to this day.
Oh, that's Yeah, no, that's actually, I've often asked a few of our surgeons if I could shadow them, and they always said yes, but they never followed up on it. And so maybe I should have done that too, because I would love to do that. Yeah, for me, it's an abstracted sort of thing. You image it, but it's something different actually seeing brains. And you still do that. I mean, it seems like you're still doing this sort of hands-on sort of anatomical studies as well. Along the way, were there other sort of, significant collaborators or people that were really helpful as far as, helping your career and helping focus it in some sense or guide it?
There was plenty of people along the way who have been instrumental in my path to this day. And that could be either directly or indirectly. And some of them have actively promoted my work. Others have just set good examples of research integrity, compassionate leadership. And others influenced my path very critically, and I never actually met them in person and they're far removed. So it's a plethora of people who stirred me into the right direction along the way and led me to where I am today.
One last thing about that caught my attention, and I was really intrigued by this, is that so you actually live on a houseboat. Yes. And I always think of that as a really cool thing to do. Is there a story behind that, or is this just something you just? It's a real conscious decision to say I want to live on a houseboat, and there's probably all kinds of issues.
Oh God, yes, it's very interactive living with the elements. There's a story behind it that is very long if we go into detail, but the short version is that I, when I was little, developed a fear of water. But that really annoyed me because I love water. It's really my element. So I was trying, being a psychologist as well, trying to overcome my fear using exposure therapy, basically just putting myself in a body of water of some sort. And at some point I realized that I'm perfectly fine being in water when there's something that separates me in the water. So I started kayaking in London. And when we did kayak on the Thames, the path usually goes past houseboats, and they're stunningly beautiful. So I was sitting on my kayak paddling along. It was like, one day I want to live in a houseboat. But obviously I could never afford them in London. And then I moved to Bordeaux after Brexit. And Bordeaux is close to the Atlantic, right? So I was like, great, I'm going to learn to surf, pushing it even further. But then COVID came about and surfing was not an option anymore at that point. And then once COVID lifted, I moved to the Netherlands. Now in the Netherlands, they have plenty of houseboats. And I got very lucky. It was sheer coincidence that I ended up on a houseboat, and I love it. I can't imagine living anywhere else anymore.
Well, that's great. That's cool. So you just sort of like, if you have a car or something, you just park at the dock and just, that's awesome. That's very cool. All right. Well, all right. Let's get into the science. So as I, was sort of reviewing some of the work that you've done, it seems that, in recent years, there's been sort of a theme that I sense developing, and I feel it developing in the field in general, and that is, brains are different. They can't necessarily. I mean, sure, it's okay to average them together for certain purposes, but they're functionally and structurally different between individuals, between species, and we can learn about them, mechanisms of evolution from that. And these differences are reflected in behavior and also pathology. And also, you mentioned also that the regions, and we'll talk about this, that are most different, that have the most variability, are potentially the most recent evolutionarily. And really, I just sense that that you're sort of, all these papers sort of suggest that we've only really scratched the surface on this unique relationship between behavior and in particular white matter structure or tractography. But so is that a fair sort of assessment of?
Oh yeah, absolutely.
Okay.
Yeah.
Okay. So just to go into tractography for those who aren't aware of precisely what it is. So what is MR-based tractography, and have there been previous methods, and how is this better?
Well, that's a lot of questions in one. Well, a nice link to what we've just talked about, diffusion imaging is based on water molecules. So it kind of makes sense that I ended up studying diffusion imaging. But the beauty of it, and that's why it has such an impact, is because it's really the only method that we have available to study the white matter connections in the living human brain. There have been other methods previously where you can study the white matter or the connection anatomy in the brain, but they were limited to other species, for example, axonal tracing, or postmortem brains, Klingler postmortem dissections. And you alluded that I still look at brains, so actually every single year, part of a workshop where we dissect the white matter of the brain. But these two methods that we had available have their obvious limitations. One is that axonal tracing studies in animals show us a beautiful map of what is connected to what in the brain, but it is the animal brain. And as you already said, there is a huge degree of variability between species. So we can't just take tracing studies and map them onto a human brain one-to-one. And the same goes for postmortem. Looking at the brain after the person has passed away will alter the anatomy compared to what it is like in the living brain, and especially when we consider combining it with functional imaging, the activations of the white matter. So that's the beauty of tractography, that you can study white matter connections in the living human brain, and that obviously comes with the added advantage that you can not only study it once, which is the case for postmortem dissections, but you can repeatedly study it and also see the changes across the lifespan. And then what is it? As I mentioned, you use water molecules, and you very crudely describing the method, right? The idea is that if there's nothing that hinders the movement of the water molecule, they can freely diffuse in all directions equally. And that signal you can pick up as a sphere. Whereas if they propagate along an axonal structure, they should be hindered in the perpendicular directions, but they can easily travel along the axonal structures, and that you pick up as a ellipsoid shape. And that's how you differentiate whether you're in the white matter or you're not in the white matter. Very crudely put.
That's perfect. And certainly diffusion imaging and other types of diffusion imaging have, they get these measures like what's called fractional anisotropy, but it's the step of actually taking that and actually building tracks that's really interesting. And I feel it's been around for at least 15 to 20 years, but it hasn't quite caught on. And I'm not sure whether it's because of the processing, whether it's so variable, or because people feel like there's enough information in fractional anisotropy, or what do you think about that? It seems like it's catching on more now, but especially with your work.
So there's a couple of steps along the way, I would argue. First off, diffusion imaging has been around for a while, so we're actually born in the same year. Tractography came a little bit later now. Yeah. But the early days, and I still remember seeing the first tractography reconstruction about 15 years ago. It took ages to get the reconstruction. So you would draw a region based on best anatomical knowledge, and then you had to have a coffee or two or three and wait for it to reconstruct on your screen. And then the first time I saw it and rotated on the screen, I was like, where am I? I completely lost orientation because there was a lag in the visualization. So we have come a tremendous long way, because now you place your region and instantaneously you can see the white matter. But the pre-processing steps still take some time. And then it depends on the sequence that you have, how many steps you need to implement to go from a raw DICOM data file to a whole brain tractogram. And that again then depends on the scanner that you have. So in the clinic, we still most of the time have 1.53 and very short diffusion sequences. Whereas on research scanners, we're going crazy. We have all the p-values, all the gradients, all the directions, and you can get beautiful reconstructions because you have the computing power to analyze it, but also the time to acquire long scans. In the clinic, you don't have that. Now, in terms of the impact, I think the complexity in the pre-processing does play a role, but there is quick and easy solutions that are scanner implemented that allow you to reconstruct it in the clinic, with the caveat that they use older algorithms. So the crossing of fibers, for example, is not as well resolved as you would see it in beautiful research, high-tech acquisitions.
Yeah.
And then in the general clinical population, tractography or white matter studies have caught on primarily in the neurosurgical community, actually, because in stroke, the damage is already done, for example. But neurosurgeons have to go in and actively damage brain tissue by cutting through certain parts. So they were very much interested in where the white matter is in an individual patient. That's also relevant, to avoid relevant critical pathways when they try to reach the tumor.
Yeah, no, actually, you're absolutely right. I feel that, and for that purpose, it's a little bit more straightforward. They could, instead of making an entire tractography map, they could probably draw two seeds, from one area to another, and say, okay, these fibers go through. I want to make sure I don't cut these fibers that wrap around some tumor or something. Yeah, and I've seen that done, and that's wonderful. I maybe this is getting ahead of myself a little bit, but I'm always thinking about how do you actually make a step from the research applications, like you mentioned, the algorithms that, and also the pulse sequences that really nicely solve, or at least address really well, the problem of the ambiguity between crossing and kissing fibers and where things ultimately go, and bring that to daily clinical use. I mean, it seems that this information is really useful for a lot more applications. It would be nice to either have a third-party vendor or something, or some vendor just developing this. And that seems harder. I mean, it seems like,
Well, the thing is, once you put it on the scanner per se, or have a plug-in software, it has to get FDA approval.
Right.
So that's a whole different ballgame in that case. But clinics are catching up. They're getting better scanners every day. And there is more and more awareness and interest in actually mapping the white matter connections, because we have shown that it's clinically relevant, right? And oftentimes, so I just had this example recently, for example, where I was approached with a clinical sequence that they had, and it was so close to being a good research acquisition, and they just had to tweak a few sequences or parameters in the sequence. Yes. So we discussed it, changed it, and now a standard clinical protocol does actually acquire good research-ready data as well. So I think that gap is closing as we move forward. But obviously there's always more one can do.
Okay. Yeah, well then, along the lines of the research, so in terms of the limits, I mean, just from reading the papers, it seems that there's been sort of 26 tracks plus a few more recently identified. And do you think there are more, or do you think that, and also just to back up a little bit in terms of interpretation, so white matter track, so, and this is one thing that sort of has struck me when I read a lot of this work, is that, there's an implicit assumption that the wider the track, the better the connectivity. And maybe that's absolutely true. Is it just simply more axonal pathways as far as that's concerned? And, is there anything potentially that it's missing or misinterpreting as far as that's concerned?
So none of our neuroimaging methods are perfect, and tractography is no exception to that rule. So there's a couple of limitations that we're very aware of, a couple of limitations that we can relatively easily overcome, and a couple of limitations where we haven't found a solution yet. So the things that we know is that when fibers cross, for example, FA, as you mentioned, drops in that particular region. Now, that doesn't necessarily mean that there is less integrity in the fibers. It's simply a crossing. And the early algorithms, especially the one that I described earlier with the sphere and the ellipsoid, they would only be able to go into one direction per voxel. And then in places of crossing, the algorithm wouldn't know where to go. So it could either decide to stop and say, well, two ways, can't choose, I'm staying where I am. And that would create a false negative reconstruction, meaning that it doesn't show a connection when we know it should be there. Or it could take one of the two and ignore the other, or average the signal and go in a random direction, creating a false positive, meaning it's showing a connection where we know there shouldn't be a connection.
Okay.
Now, this has partially been overcome by ramping up those p-values and, and directions and throwing new algorithms at it that can actually resolve the crossing much better. So now that is less of an issue. What is still an issue, however, is that we don't have a gold standard. So neuroanatomy is one of the oldest disciplines, but we don't actually know the anatomy of the brain very well. Because, as I mentioned, Klingler postmortem dissections, you scrape away the cortex and the brains themselves have to go through a freezing and thawing process over the course of about 3 months.
Wow.
And obviously, if you freeze water, it expands, and if you thaw it, it reduces again. So that could also artificially induce some artifacts. But it is the closest match to what the white matter looks like in the brain. But that means that we don't actually know 100% the true anatomy.
Okay. And yeah, I was wondering that as far as where they terminate, like for instance in gray matter, it's like I'm always intrigued by how precisely how do they terminate, and how is it inhibitory or excitatory connections that happen, or does that happen more in the cortex? And I don't know if there's any insight or any I don't know the field well enough to know whether it could be both, right? I mean, I imagine.
Yeah, so in terms of the cortical terminations, we're getting closer and closer to the cortex, both in the postmortem world and in the in vivo world. So postmortem, for example, what you can do is nowadays you can just have a series of photographs that you can then digitally reconstruct to actually have a 3D version of your postmortem brain and trace it back. We also have new methods like PLI that give us beautiful reconstructions or visualizations of the white matter, I should say. But then also with neuroimaging, the algorithms are getting better and better to get more signal out of the images that let us then track closer and closer to the cortex. So we're getting there. We're getting there.
Okay. So right. I mean, as far as and once again, the implicit assumption is that the larger the tract, the stronger the connectivity between the areas that it connects, and that's seems like it makes sense. Just along the lines of, I know that, and this sort of a tangent a little bit, but I know that with fMRI, we've been trying to look at activation in white matter. I mean, people have tried to do it with varied success. Some people that claim they see it. Have you even thought about doing that? Or I'm not sure if it's even possible for a number of reasons, possibly that, the white matter is so active for so many different things that it's hard to differentiate that, or even the vascularization is very different as far as the neurovascular coupling. Yeah, like I said, it's a slightly tangential question, but I'm kind of curious if you've thought about that.
Well, yes, and we actually came up with a functional atlas of the white matter recently. And Victor Noas, a PhD student in the team, developed a tool called the functionectome that lets you combine fMRI and white matter data and reconstruct the white matter networks observing functional activations. So that is a good proxy of the function of the white matter.
Yes, yeah. And that's amazing. That's actually, I saw that, and it's these beautiful, I love the way you graphically depict it in your papers, where you have, the various functions and you show it along the white matter tracks. And I mean, that would be a great guide for if you're trying to do fMRI of the white matter. That would be interesting. But yeah, no, I think that was remarkable. Let's just go into individual differences then right now. So you mentioned in your paper in Brain Structure and Function that, over the course of the last 15 years, there's been over 300 studies of human adults showing significant correlation between white matter tracts and cognitive measures. And it seems that in general, what we're finding more and more with fMRI as well is that we try to parcellate a brain, we try to average brains together, and there are problems with that because the brain is different, individuals have different characteristics, and so it seems like in general, it seems that the entire field is kind of regrouping a little bit to refocus on the individual differences, which is really, there's enough sensitivity to do that now. And you've been doing that already. And so one general question before we get started. So you see a lot of differences in the white matter tracts between individuals, and how much of it is due to purely genetics, or maybe due to early stages in development, or maybe just behavior as they get older, or how much is random?
The answer is everything above. So it is a mix of environmental factors, genetic factors. And Sanja Budisalevich did a beautiful study a couple of years ago where she tried to disentangle it and could show that it depends on the white matter tract if it's more genetically driven or more environmentally driven. But we're now getting those data sets that would actually allow us having the right amount of participants, subject numbers, to actually disentangle it properly. But there is preliminary evidence that shows that some of the tracts are more reliant on genetic factors. That would be, for example, the long segment of the arcuate fasciculus, whereas other tracts like the posterior segment are more influenced by environmental factors. And Michel Thibault-Deschotte nicely showed that with literacy. So learning to read actually changes the posterior segment of the arcuate fasciculus, so the parietal temporal connection.
Wow. And it doesn't matter when you learn to read. It's I imagine maybe it's early on, but so that's fascinating in itself, right? And that seems like it suggests a pretty high degree of plasticity. And does anyone know what the mechanism for that is? Is it just simply with more use, there's some sort of stress, and then it sort of becomes there's more connections? I mean, how does that even work?
It goes back to use it or lose it, right? So the exact mechanism aren't fully understood yet, but obviously if you need to integrate the functions of two areas, they have to be connected for that emergent function to perform. And we know that there's plasticity in the brain, especially in the younger brains. And then what we mean by plasticity also changes across the lifespan, right? So plasticity in a newborn brain is slightly different than plasticity in an adult brain, which is different to plasticity that we can see after a stroke, where, for example, the white matter doesn't regrow. So there's no structural plasticity in the white matter in a sense that you can regrow your connections at that point. But we do see functional plasticity where other areas compensate and take over. So there is this change across the lifespan.
Okay. Yeah, no, I was also, I mean, you brought up plasticity in terms of connectivity. It seems that you mentioned in also one of your papers something interesting about the connectivity in visual cortex with primates, and there if it's broken, it heals, it becomes more connected, and in humans it's much more fragile. That's also interesting in itself. Yeah. So okay. So you're right. I mean, I think that this seems to me. I mean, this to me it seems wide open in terms of there's an actual neurocorrelate of. I mean, people have tried to look at gray matter density and other neurocorrelates of behavioral changes or learning. For instance, learning to juggle. There was that famous paper. But looking at the white matter tracks themselves, it seems another area that seems very rich as far as exploring this. I keep on thinking of, I read a paper a while ago of, musicians have, for instance, apparently a larger left planum temporale, and I'm sure that there's white matter tracks that are correspondingly larger as well with that use. And so that's actually, if we start to understand the mechanism of that, it seems that it would be wonderful to maybe then potentially try to precipitate these changes or allow these changes to happen more easily potentially, or who knows.
But that would be the goal, right? Once we understand fully how it all works, we can also go in and fix it when things go wrong.
Yeah, exactly. And which reminds me a little bit of, and so I'm totally jumping ahead of myself, but I really have to ask this question in this context. And that is, so there was, you mentioned an experiment where visual inputs were redirected to the auditory cortex. And after they were connected, it changed the cytoarchitectonic properties of the auditory cortex to start to resemble a visual cortex, which was mind-blowing because it suggests that it's the connections that determine the structure of the cells and not, somehow the structure and everything sort of laid down. It seems like there's this causality in that regard. I don't know if you've done any follow-up experiments or anything like that, but what does that suggest? I mean,
so just to clarify, I didn't do that study.
I did that study. You mentioned it in your paper, in the wonderful Science Review article.
Yeah. So yeah, it's absolutely mind-blowing how that actually adapts in the brain. And we also see it as another study that we refer to, that the emergence of the visual word form area, for example, can be predicted based on the connectivity before it actually shows up in functional imaging. So there definitely seems to be a causality in that sense, that white matter determines where function can emerge in the gray matter or the cortex. And that also then has implications for plasticity when that white matter is severed.
Yeah, and it seems that I'm wondering, right, there's just probably a critical period for this, but regardless, it really is interesting. It sort of hints at a mechanism for determining the cytoarchitectonic properties that's new to me. And so going back to differences, when you have white matter tracks, it's one thing saying, oh, they look different because you can just see it. Is there a way now for I'm sure that's for characterizing how are the differences characterized? Because, right, I mean, in terms of fMRI, you have center of mass, and it's very simple calculation. But here it's like you have this complicated structure, and that has different tortuosity and thicknesses and all kinds of things. And what is the current standard and how to do that?
Yeah, so fundamentally, you're absolutely right. We know there is a huge degree of variability, both in the white matter and the gray matter, by simply looking at brains.
Yeah, our brains can just process it and say, oh, it's different.
Yeah, it's super different. And you also, in the beginning, you alluded to the fact that it's not just my research that looks into individual differences, but it's the entire field that's regrouping around the topic. And that is because in the beginning, the interest was more in regards to how does the brain work in general? Which part does what function? But now we actually have the amount of data and the computing power and statistical tools to start going backwards again. It's like, okay, we have a rough idea of what happens where. How can we now study that in the individual brain? And using the white matter is one avenue to explore those variabilities between us. And tractography, for example, for the most part, at least, and especially in the clinical studies that I published, but most of the time I stay in native space. I never go into a standard template space.
Okay.
And what any tractographer should be doing is go through that process of learning the anatomy properly. And most the work that we publish has been manually dissected, which does take its time. And you sit there dissecting 200 brains, all the white matter tracts.
Wow.
It's quite labor intensive, but it is worth it, especially if you're interested in variability, because an atlas won't give you the results that you need in that case. But that means it takes time. And then once you have your dissections, you can extract many different indices from that. So you can just take the size of it, thickness of the tract, bigger in one person than the other. You can extract indices along the tract. So that could be the fractional anisotropy that you mentioned, or any other index. There's plenty of them depending on the algorithm that you use. But FA, and we've shown that in the brain extension paper that you mentioned, is by far the most commonly used index from tractography studies, with about 50% of the studies just looking at FA.
Wow.
So you have all those measurements, and then you can use them in statistical models. And these days we have nonlinear models as well. And you can weight them, see which one is more relevant or explains more variance in the difference. And yeah, that's the power of the current tools that we have.
Yeah, no, and also I imagine that if for some reason people want to average, I mean, it seems like the problem of, making an average white matter track atlas. I mean, certainly it can be done, but if you're trying to do group comparisons or something like that, it seems like a huge problem as far as that's concerned. Even more so than putting things into a standard anatomical template for looking at, for instance, gray matter activation and that sort of thing.
Yeah, yes and no. So we did this study with Paula Croxon a couple of years ago, where we looked at the variability in the white matter across the brain. And what we showed in this study is that the variability is not homogeneous across the brain. So there's areas where we have white matter that is more alike in terms of its FA and shape between us, and areas where there's more variability between us. And what you can see in those maps is a gradient. So the more central structures, the older parts of the brain, are more alike, and the more recent structures tend to be more variable between us. Now, what that means is that if you want to postulate the white matter with an atlas, then probably if you stay in the center of the brain, that's perfectly fine, because we're more alike in the center of the brain in the older parts. But if you go to the more recent parts, then a postulation based on atlas may be less reliable.
Okay. And so that's actually, so that's actually interesting in itself. And let's maybe talk a little bit about that too. So the evolutionarily older areas are much more similar. And yeah, I remember seeing those images where you're looking at the differences, and it seems that, like, for instance, between primate brain and human brain, and also among human brains, it's like, visual cortex, prefrontal are much more variable. And that, it almost seems to suggest that sort of nature is still in the process of figuring out. I was surprised how much of the visual cortex is so variable, and there was some prefrontal as well, but motor seemed like it was pretty well behaved. And
Are you saying variability is misbehaving?
Oh, well, actually, right. That's I would agree. I think that maybe the variability is the good thing. That's the most interesting. Yeah,
But
yeah. So
with the visual cortex, for example, if you go back and look at individual brains, that has been shown in postmortem brains just how variable the visual cortex is between us, but also between the left and the right hemisphere in the same individual. So initially, we were also surprised by how variable it is, but actually, when you look into it, that variability has been documented using other methods.
that's interesting. And another thing that you mentioned in the species comparison was the increased laterality in humans versus primates, and I was really intrigued by the potential explanation for that, in the sense that once the brain gets big enough and the information transfer in the corpus callosum gets high enough, there's a limiting factor of the size of the corpus callosum, so it might require then more of an efficient transfer, one side or the other, rather than relying on left to right hemisphere. That it seems like it was solving a structural information, problem in that regard.
Well, what you also see in this study is that the areas that tend to be more lateralized in the human brain tend to be those areas that we associate with higher cognitive functions in neuroimaging and lesion studies. So, for example, the language areas, roughly. So, yeah, there's definitely something interesting there.
Yeah, and right. It seems like there's somehow a principle of it's sort of the brain is solving this problem of efficient information transfer with limited space and of the white matter tracks. And so then it made me think, can we? So now we're studying evolutionary differences, looking to the past. Is there any possibility of projecting to the future? And, looking, maybe 500 million years in the future, what would, I can almost imagine, right, what would the brain evolve towards in that regard? If it was getting bigger, then you would have maybe some more specialized functions or something like that. Have you tried doing that?
Projecting forward, not that far yet. But also the question is, so at the moment we take a snapshot, right? We see those differences. Some of them were mapped for the first time. If we look at the language network, for example, we see that the arcuate fasciculus in the left hemisphere is quite considerably sized, but then when you look at the right hemisphere, the less dominant hemisphere for language, then the arcuate can have many different sizes in the in the right. Now, the question is, what is the more evolved brain? Is it the one that is more bilateral or the one that's actually highly specialized with the left side? And that we don't know yet. Now, when you correlate those measurements with behavior, what we tend to see is that people who are more bilateral tend to be better on language, verbal learning, memory tasks. And also, if you have a stroke in the left, but you do have a strong arcuate in the right hemisphere, you can compensate better for the loss of the left. So that might indicate that a more bilateral network is actually more advantageous in that particular example. But it is a snapshot.
Yes. So you're right. So the bilateral is more robust to injury or whatnot. But then that's so right. Who knows what this could be an intermediate stage, and somehow we'll figure out how to make it more bilateral if right, losing language is something that affects our survival in that regard. And along those lines, actually, I was sort of just curious about language in general, and there was some discussion in your papers with, it always intrigues me with language in general, sort of written language, that the brain didn't evolve to read, and so this is almost an off track of the white matter track question, but what were those areas primarily? What was their evolved function before we used them for reading? I guess that's nobody really knows.
Yeah, I was just gonna say, no, I need to speculate.
Sorry, I'm asking you like impossible questions. It's
It's all about the future and the past. Well, one thing that's clear is that language didn't just randomly emerge, right? There wasn't a single accident in nature that then made us talking creatures or reading creatures, but it slowly evolved over time. So it's probably fair to assume that those areas probably subserved some related functions of articulation or communication of some sort, and then specialized and repurposed through evolution to create this amazing linguistic brain that we have nowadays.
Yeah, no, I think right, the verbal part, but then right, associating the visual, and you're right. I mean, it could be, that's a little bit of plasticity in development allows to, use this system for the visual word form area to connect it with language comprehension and talking. And so it's amazing. It's truly amazing. And I think that, this is just starting to work on that. So as far as sort of another tangent a little bit, you look at mostly white matter tracks, and are there any other sort of measures that seem like they could be sensitive to behavior, like things like, I mean, of course, people have tried looking at gray matter density, overall brain size, maybe gyrification or surface area. Is there any hints that maybe white matter is the white matter tracks are the most sensitive, or have you been looking at everything?
Well, we're just beginning to look at everything, right? Because most of the time, again, because we had to collect our own data, which takes time, most people specialized in one method, in fMRI or in diffusion tractography. But now we have this beautiful playground of data sets that have it all. So we're only at the cusp where we can actually combine all the rich data sets and all the information and try to model and assess which parts of the data are more predictive of our behavior. And there is preliminary evidence that suggests that the structural connectivity is a reliable predictor of behavioral differences. But you just listed a range of methods that also have shown variability, and that variability in those measurements actually correlates with behavioral measures and functions. So there's a lot out there to study for variability.
Yeah, and just to mention, right? I mean, you're in your paper on in brain structure and function on white matter variability, cognition, and disorders. You have these beautiful charts where it shows that even the white matter tracks that are not what you would think would be associated with disorders, they have there's studies associating them with psychiatric and neurologic disorders. And I really get the sense reading this paper that we're just at the cusp of really, starting to dig into this, which is just, it's a really nice way of describing it. Yeah.
That study actually surprised me in multiple ways. The first one was that it was supposed to be a pet project for a weekend. And then it took a year to put it together because there was a lot more studies out there than we initially thought there was. So that was surprise number one, positive surprise. But the other surprise was that most of what we know about the function of the white matter, we actually know from clinical populations. So the majority was neurological and neurosurgical patients, then psychiatric patients, and only about 25% of the studies look at healthy controls. And then the third surprise was that we always consider certain tracts to be associated with specific functions. So the arcuate, for example, is that classical language connection.
Yes.
But when you look at the literature, then you can see that there is no one tract, one function association for any of the white matter that has been identified. And there is a pattern where the variability in the anatomy is associated with the variability in cognitive profiles. So that was quite an interesting finding.
You can use this to start to generate all kinds of hypotheses as to what's happening. And also looking at this, it jumps to mind, and maybe we'll jump into the final area that I want to talk about is emergentism. It's important to understand, I think, that you have the white matter tracks, and that does show what areas are connected. But with fMRI, you have measures of connectivity that may be multiple white matter tracks away from each other, and the entire network is somehow doing a function in that regard. So it's right, and as far as that's concerned, it's just always intriguing to understand, what's the difference between networks that are connected by single white matter tracks versus more distant networks, and will they eventually have a track connecting evolutionarily or what? So that's
yeah, and it's quite interesting because there is a link quite often anatomically and functionally speaking, but also cognitively. Like language is one cognitive functions, but we can't talk without memory being linked in, top-down modulation, executive functions. Like everything we do is connected in a way and has to integrate at some point. And we recently applied the functionectome that I already mentioned to resting state, for example, and were able to identify that there's quite an overlap between the white matter components and resting states. And the hypothesis that then comes out of that is that if a lesion damages a overlapping area in the brain, that would have an impact on a lot more functions and then return a more diverse clinical presentation than if a lesion is only overlapping with one structure and function.
Yes. Yeah. That's right. I mean, that's also, that's really, and so you can actually start to do sort of lesion symptom mapping where it's more of a network. I know that, but also you can imagine doing the opposite. You can imagine, if you're trying to enhance networks or suppress networks, you can imagine trying to do neuromodulation where you're getting at these areas that are sort of the hubs that affect these behaviors. So,
which many people try to do. So there is a lot of work being done using TMS, TDCS, or even direct stimulation during awake surgery.
Oh, wow. It really is something where we're just at the edge of more deeply understanding this. I mentioned a couple of times sort of the essence of emergentism, and that is the idea that we're moving away from, and it's a good thing that, it's not just, one area, sort of one function. It's really the interplay of multiple overlapping areas that emerge with these functions. And I don't know if you wanted to expound on that a little bit, and because you sort of made it it's kind of what you're doing now. I mean, it's pretty much the essence.
Yeah. So the idea really is, as you said, that there is no one area, one function association, but also there's no one track, one function association, as I just explained. But really, the complexity of our behavior emerges through the interaction between areas. And the example I always give in lectures is that asking a neuroscientist where language is in the brain is similar to asking an orchestra where their song is. It's nowhere. You can't find it. It's not in the individual instrument. It's not in the notes. It's not in the conductor. It emerges through all of them coming together in a synchronized dynamic pattern that the song emerges. And that is how I see language in the brain.
Yeah. And it's interesting because people, when they hear this, when they hear you say that, and I imagine be like, well, of course, everyone knows that, there's a connection. But there really is sort of this nuanced idea that to really understand how you perceive music or how you produce language, and to really understand how you produce language, you have to understand the network properties as opposed to just simply studying any area in isolation. It's really this interplay, and that's where the information is. And it's so hard to do that with studying each region or parcel independently. And that's really hard as well, because I imagine, we're looking at one scale here, and that's another question I've had, is in terms of looking at we have very high resolution, you have very high resolution for looking at white matter tracks, but one wonders how, across scales, what the interaction is. And if there's a scale that's like most relevant in some sense, and hopefully it's the one we're looking at now, but maybe there's other finer scales or that might give this information.
But that's another theme or topic that our community is regrouping around, right? Is the granularity of how we look at the structure and the function of the brain, and trying to bridge those gaps that we have.
Yes. Yeah. And I totally agree. I think that the field is regrouping in that regard. We talked a little bit about your stroke disconnectome, but why don't you talk a little bit about how, using sort of a sense of how connections are broken and how behavior is interfered with. I mean, it's sort of going back to the old lesion studies, but now we have much more insight into the connections. You can start to understand, what's important as far as that's concerned.
Yeah, so the clinical lesion studies is very close to my heart, and something that I've done throughout my career. And as I mentioned in the very beginning, that's also where I started initially. And obviously, we're learning more and more about how to study lesions in the brain, and that lesions, for example, aren't normally distributed in the brain. They tend to cluster in specific vascular territories. So that then also means that when we look at lesions, we tend to be slightly biased in which parts of the brain are affected, where we have the highest effect sizes when we then do group analysis studies. And really the idea with the disconnectome that Michelle developed was to make it clinically applicable, because as we discussed, the pre-processing pipeline takes a little while. The clinical scanners often don't have the resolution that you need to really look at those fine anatomical features. So what you can do now is with a disconnectome symptom discoverer web app, you take a lesion, you upload it on the web app, and you get the pattern of disconnection. So you go beyond the lesion, because we know a lesion affects the focal part of the brain that is damaged, but it also has a knock-on effect to other distant brain areas. And that you can capture with the pattern of disconnection. Now, Leah Talozzi recently ran a study where she used that pattern of disconnection as a predictor for recovery. So using the acute lesion, the pattern of disconnection caused by the acute lesion, to predict long-term outcome 1 year after the stroke. And she had fantastic results with reliable predictions for long-term recovery. And that is something that we've been absolutely missing in the field, because at the moment when a stroke patient comes into the clinic presenting with whatever deficit, the first question is, how will I be in the future? Will I be able to talk again, walk again? And most of the time, we don't know.
Right.
Whereas now we're getting the data that reliably predicts the future, as you wanted in the beginning. A bit shorter term with a year.
It's a shorter term. And just using that information about where this stroke or lesion lies on the white matter tract connections, and that would be right. And that information is incredibly, extremely useful as far as that's concerned. And I always wonder. I mean, it is interesting how, if a track is completely broken, or if it's partially broken, how much of the function is compromised. Let's say if it's only partially, if you break half the fibers, is it like an electric current where that's okay? It's just half the fibers, you still are communicating, but in this case, it seems more of a continuum. Interesting. And I think that, right. I mean, anytime you have a better prognosis as far as likelihood of recovery, and that could be profound impacts in clinical practice.
Yeah.
Some final questions. Well, first of all, just to shift gears completely, you really have been so engaged recently with OHBM, and really been helping the meeting in so many ways in the society, from everything from the working with ComCom to now the technology task force, and the program chair, YouTube manager for OHBM and whatnot. How do you actually balance? This is a lot of work, and how do you balance all this? I mean, just obviously maybe sleep is compromised, but
yeah, I was just going to say I'm probably not the best to advocate life-work balance at this point. But the big advantage that I have is that I love what I do, both the science administrative side, but also the research side. So I happily volunteer extra hours to try and fit it all in. But you are right, sleep is a little bit compromised these days.
And definitely your passion shows through, and it really, I think, helps every aspect of the meeting as far as that's concerned. So, and it's clear that you love it, and I think that's actually a great thing. I mean, to do what you love and put your energy into it, and sometimes things get unbalanced, but that's maybe okay. Who knows?
Also, HBM is quite a rewarding community, right?
Yes.
You can give a lot, but you also get a lot back, and it's quite enjoyable working with all of you.
Yeah, I mean, it's I think, it's a unique community because we have methodologists, we have neuroscientists, we have clinicians, and we all realize we actually have to communicate a lot to advance the field, and it's really kind of a community, and yeah, it's a lot of fun. I agree. It's so fulfilling.
It's in the end, it's the integration, right? We're all more together than the sum of our individual achievements. So there you go.
Exactly. The emergent properties of our community. It's moving it along, but yeah. And so, do you have any? I know that it's still probably pretty early, but do you have any insight as to how the meeting coming up will turn out, and is there anything unique or?
So the committee is working very hard to put the meeting together. We're now entering the hot phase. Now that everyone submitted their great work, we have to sit down and sort it all out. So that is coming together very nicely. There's a lot of work going on in the background. There's also a nice integration between committees. So the ComCom committee and the executive committee are integrating further and further. So there will be a ComCom digest, for example, that comes out, highlighting all the changes that we're going through. A new blog is coming. So that's quite exciting. But also the biggest change, I would argue, is that you will have OHBM 24/7 all year around. And that is because we're going through the symposia submissions, and the ones who aren't submitted for the in-person meeting will be offered to have an online version of their symposium. And that means that there is OHBM content throughout the year. So we're not limited to the one time per year in-person meeting, but you have all area access all year.
That's great, and that makes perfect sense too, as far as adding more content, and not necessarily. I mean, sometimes people just want to receive the content. Of course, there's advantages of the meeting, but that's a great idea. That's a great innovation, and I look forward to having yet another thing that I can download all this information, and it certainly helps people who can't attend the meetings, in other countries or whatnot. They'll always have that, and so that's a wonderful thing for dissemination.
It makes it a lot more accessible to everyone.
Yes. So the final question: where do you think this area that you're working on is going, and so what do you find most exciting, and as far as what you plan to do in the next few years?
There's many things that I find exciting about the work that we're currently doing, which is good. It means we're doing the right thing. But one of the things I really want to get to the bottom of is the magnitude of variability. How different are we? So genetics, they have their magic number that we often can see, where they say that we're 99.8% alike. And that basically means that there's a 0.2% uniqueness factor in our genome that is sufficient to give rise to a wonderfully complex diversity amongst the species. So I kind of want to know what our brain's magnitude of difference is. So that's one of the projects. And then akin to genetics, where that variability has been linked to diseases and changes throughout the lifespan, I want to know how our variability actually relates to behavioral changes, but also most importantly in a clinical setting. So right now, when we look at clinical demographic data, we can predict about 30 to 40% of the recovery. And I want to close that gap bit by bit. And I think variability is playing a very crucial part in explaining that remaining variance and recovery. So hopefully we bring it back to the clinic and have a meaningful impact longer term.
Yeah, that sounds, that's really right. That sounds, it just made me think in terms of genetic variability. How does that translate to something physical? And if the transformation of measuring brain variability, does that tiny little bit of variability amplify in some way in certain areas or in different ways? And so that's, and also trying to look at how the variability is linked to behavior or other measures. So that's a really exciting work. And well, thank you, Stephanie, for doing this podcast. I learned a lot, and I'm sure the whole audience will be keen to read your next papers and follow the work, and hopefully we'll be seeing you at OHBM. So
for sure. Thank you so much for the invitation.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Ömer Faruk Gülban and Jeff Mensch.